Welcome back to the deep dive. Today we're looking at one of life's really great paradoxes.
It's a fascinating one.
To live a long and healthy life, your cells have to constantly respond to stress. But the very system for handling that stress, it weakens as you get older.
Right. The toolkit gets rusty.
And we're diving deep into one specific part of that toolkit. Translational regulation. So that's the process where your cell reads its genetic instructions and actually builds proteins.
And here's the part that it always blows my mind. This single process, translation, can use up to 75% of a cell's entire energy budget.
75%? That's incredible.
It is. I mean, think of the cell as a city, the translation machinery. That's the most expensive, non-stop factory running the whole place. So when a crisis hits...
It's like a power outage.
Exactly. A power outage or a supply shortage, that factory can't just shut down. It has to make this incredibly precise pivot. It slows down global production to save energy, but at the same time, it has to ramp up production of, say, the emergency repair crews.
So we're focusing on what happens after the gene is transcribed. We always hear about the genome, about genes turning on and off, but this is the immediate response.
It is. It's both the sensor and the rapid responder. It's what allows the cell to course correct in real time. We're going to unpack how it handles three key challenges of aging: misfolded proteins, oxidative damage, and nutrient sensing going wrong.
Okay, so before we get into any of those specific stressors, let's talk about the shared core. The two main control pathways the cell uses pretty much universally, the accelerator and the emergency brake.
Let's start with brake. It's called the integrated stress response, or the ISR. And it's just this brilliant, elegant pathway that's all about shutting down global protein production when things go sideways.
So how does it do that without just destroying everything?
Right, it's not a wrecking ball.
Okay.
It targets the starter motor for every single ribosome, a little factor called EIF2.
Okay.
Normally, EIF2 kicks off the process. Then it gets recharged by a recycling factor called EIF2B. But under stress, the cell sticks a chemical tag on EIF2.
And that stops it?
It does more than that. It actually converts EIF2 into a blocker. An inhibitor for its own recycling factory.
Wow.
So it gums up the whole works instantly. The entire assembly line just locks up.
That sounds like a universal shutdown button. But the genius here is the sensing, right? It has multiple inputs.
Absolutely. There are 4 specific kinases that can throw this brake switch, and each one is like a different kind of smoke detector. You have GCN2 for nutrient starvation.
So no amino acids.
Right. PRK for unfolded proteins in the ER, PKR for viral RNA, and HRI, which senses trouble deep inside the mitochondria. It's a highly targeted system.
Okay, but here's the really critical part, the thing that makes it adaptive. Global translation stops, but certain stress genes actually get a boost. How does the cell get around its own shutdown?
That is the selective genius of it. It's because of something called upstream open reading frames, or URR.
URRF.
Yeah, so imagine the genetic message for a stress gene like ATF4 is a highway. Normally, the ribosome gets on an early decoy exit ramp, that's the URRSHA, and it just falls off.
So it never reaches the main destination.
Exactly. But when the ISR is active and initiation is super slow, the ribosome often misses that decoy ramp. It just scans right past it and starts at the real start of the gene.
Yeah.
So you end up with a huge boost in this critical stress regulator, ATF4.
It's a molecular hidden passage designed just for emergencies. Amazing. Okay, so that's the brake. What about the accelerator?
That would be MTOR complex 1 or MTORC1. If the ISR is the emergency brake, MTORC1 is the growth accelerator.
Okay.
It's constantly asking, are there growth factors? Is energy high? Are there lots of amino acids? And if the answer is yes, it floors it. It promotes growth, proliferation. Stress, of course, usually turns MTORC1 off.
And when you take your foot off the accelerator, what happens to protein synthesis?
It drops instantly. MTORC1 controls the part of the machinery that grabs the start of the mRNA message. When it's off, an inhibitory protein just stays clamped on so nothing can start. And it also slows down the ribosome's movement, the elongation rate, by activating another kinase. Okay, so let's connect this to our first crisis,
Proteotoxic stress. This is the buildup of misfolded proteins. I mean, it's a huge signature of aging, of neurodegenerative diseases. Let's start in the cytoplasm.
In the cytoplasm, misfolded proteins trigger the heat shock response, or HSR. The driver here is a transcription factor, HSF1.
Right.
Normally, HSF1 is kept quiet. It's bound up by chaperones like HSP70. But when a wave of misfolded proteins shows up, they act like a sponge. They soak up all those chaperones.
Freeing up HSF1.
Freeing HSF1 to rush to the nucleus and turn on the genes for, you guessed it, more chaperones.
So, okay, the cell has hit the global brakes with the ISR and MTORC1, but it desperately needs to make more HSP70 right now. How does it make those critical proteins when the lights are basically off?
This is where those amazing gene-specific shortcuts come in. The mRNA message for HSP70 has special features built right into its starting sequence that let it completely bypass the normal rules.
Like what kind of feature?
Well, we see things like ribosome shunting, where the ribosome literally just skips over a whole section of the leader sequence to find the start. Or something called an internal ribosome entry site, an IRS.
Which is like a private landing pad for the ribosome.
It's exactly that. It doesn't need the main airport at all. And there's another layer here. The HSP70 message gets these little chemical flags put on it after stress, a modification called M6A. And these flags act like signal flares, helping to recruit initiation factors directly, again, completely independent of the cat.
So it's a multi-pronged evasion strategy. And it's not just about making new chaperones, right? The sources mention that ribosomes actually pause during this kind of stress.
Yes, this is active quality control. Ribosomes will pause at very specific spots, usually where they're making highly hydrophobic peptides.
The ones that are most likely to misfold.
The ones that are absolutely going to misfold if a chaperone isn't there waiting for them. So the pause is a temporary reversible stop. It's like holding the product on the assembly line until the quality control inspector, the chaperone, is available.
But in disease, this whole beautiful process gets corrupted. The protein aggregates you see in diseases like ALS or Huntington's, they don't just sit there. They actively break the machinery.
They absolutely do. It's molecular sabotage. For instance, the toxic repeats from the C9orf72 gene in ALS, they can physically bind to the ribosome's exit tunnel. They just create a physical plug.
Yeah, a roadblock.
A total roadblock. And mutant Huntington protein, it binds up and sequesters a critical factor called EIF5A, causing these massive damaging pileups on a ribosome.
So the big takeaway on proteostasis is the cell uses these universal shutdowns, ISR and MTORC1, to save energy, but then it relies on these incredible molecular shortcuts and special RNA features to force critical repair proteins into production. It's fighting against its own emergency brake.
It has to.
So here's a question for you to think about. Considering that huge energy cost of translation, what's the adaptive benefit of slowing everything down, while at the same time selectively boosting just one part of it, like chaperone production? Let's shift now to our second section. Mitochondrial stress and oxidative damage. The mitochondria, they're the powerhouses, but they're tricky because they're run by two separate genetic manuals.
Yeah, that duality, it makes their stress response, the UPRMT, incredibly complex. Now, in mammals, the ISR is still a central player. It drives the response through ATF4. But the sensing is just exquisite. We know that one of those ISR kinases, HRI, is switched on by a very specific sensor called DELO1, which flags when protein import into the mitochondria fails.
And the most amazing part to me is how translation itself becomes the quality sensor for the entire organelle.
It's a rapid, localized alarm system. If there's just some mild damage, a protein called BINK1 actually promotes translation of repaired transcripts right there on the surface of the mitochondria.
Okay, localized repairs.
But if the damage is severe, that local translation stalls, just stops. And that stall itself is the distress signal. It recruits this machinery for no-go decay, which ends up tagging the entire mitochondrion for destruction and recycling, for mitophagy.
So translation quality literally equals organelle quality.
You got it.
All right, let's talk about oxidative stress, the cellular rust. These reactive oxygen species, ROS, they damage everything, including the ribosome itself.
Yeah, ROS can introduce errors into the mRNA template, and they can chemically modify the ribosomal RNA, the machine's backbone. They usually just stop synthesis cold.
Usually.
What's so interesting, though, is that researchers have found some of these oxidized rRNA molecules still on actively translating ribosomes, which suggests that maybe some tolerable level of damage acts more like a check engine light, a signal of stress without forcing the car to pull over.
And the cell also uses ubiquitination to slow things down. It sounds like putting a molecular zip tie on the ribosome.
That is a perfect analogy. An enzyme called RAD6 puts these specific ubiquitin chains on ribosomal proteins. And this causes the ribosome to halt, but only as certain amino acid patterns, like XIP.
Why there?
Because that modification physically gets in the way of the elongation factor, EPF2, binding properly. It's a structural break. And then when the stress passes, another enzyme comes along and just zips it right off. Synthesis resumes.
Okay, let's talk about the tRNA response, because this seems like one of the most dramatic mechanisms.
It is. This is the fragmenting response.
Mm-hmm.
Under oxidative stress, an enzyme, an endonuclease called angiogenin, just starts chopping up tRNAs. It
Oh.
can strip off the end, which depletes the functional pool, but more importantly, it generates these little fragments, these tRNA-derived stress-induced small RNAs, or tRNAs.
And these tRNAs are like molecular shrapnel. They actively disrupt the process.
Precisely. Certain tRNAs, the ones from cysteine and alanine tRNAs, they have this specific chemical motif that allows them to physically knock the main initiation complex, EIF4F, right off the mRNA cap. It's a direct, fast-acting weapon to shut production down.
But even in that chaos, the cell has to boost its antioxidant proteins. How does it make sure something like a selenoprotein gets made?
It uses these really clever adaptive codon strategies. So the genes for these critical antioxidant proteins, they tend to use codons that match up with tRNAs that are naturally more abundant during stress.
Us, it plays the odds.
It does. And selenoproteins are the best case study. To make them, the cell has to recode a UGA stop codon and turn it into an amino acid. And it turns out that oxidative stress actually promotes a specific chemical tweak on the selenocysteine tRNA that makes that recoding process much more efficient.
So it guarantees you get your antioxidants when you need them most.
Exactly.
Okay, finally, the most counterintuitive idea here, adaptive mistranslation. The cell sometimes deliberately sacrifices quality for immediate survival.
This is the definition of a desperate measure. In acute stress, the enzyme that loads methionine onto its tRNA gets modified, and it starts making mistakes. It starts putting methionine on the wrong tRNAs.
Then why on earth would it do that?
Because you get this modest increase of methionine sprinkled all throughout the proteome. And methionine is a fantastic scavenger of ROS. It's like giving the entire cell an instant anti-rust coating. It's terrible for long-term protein quality, but it might save your life in the short term.
So the big takeaway from oxidative stress is that the cell deploys everything from molecular zip ties to tRNA-based weapons to stop non-essential work, while using genetic hacks and even deliberate errors to prioritize its own survival.
It's a battlefield. It really is. So here's the next question for you. The cell will sometimes sacrifice fidelity for survival. How might that short-term gain, that adaptive mistranslation, influence the long-term risk of neurodegenerative diseases, where protein quality is everything? All right, let's pivot to our final stressor, deregulated nutrient sensing. We already know that if you restrict pathways like insulin IGF-1, you can extend lifespan, and a big part of that is by turning down translation, right, via mTORC1 and the ISR. But amino acid sensing is incredibly nuanced. If you deprive a cell of, say, arginine, it paradoxically allows more translation to start than if you deprive it of other amino acids.
And why is that failure to shut down? Why is it a bad thing?
Because it leads to the total exhaustion of all the arginine-loaded tRNAs. So ribosomes start stalling hard on every single arginine codon. And that causes this massive cascade of problems that ultimately leads to lower cell viability.
So the cell learned that a really strong immediate shutdown is sometimes the best survival strategy.
It's better to hit the brakes hard than to run out of gas.
Let's talk about two metabolites that directly connect diet, longevity, and translation, taurine and spermidine.
So taurine levels drop as we age. And we know that supplementation is linked to improved health span. We're now learning that taurine is critical inside the mitochondria.
Okay.
It's used to modify mitochondrial tRNAs, and this modification is essential for the accurate translation of the respiratory chain components. So the health benefits could be coming directly from making sure those mitochondrial factories are running with high precision.
And spermidine is connected to this really unique translation factor, EIF5A, and a modification called hypusination.
Yes, spermidine is the essential ingredient for hypusination, this very unique and totally essential modification on EI5A. You can almost think of EIF5A as the autophagy locksmith.
Like the autophagy locksmith. I like that.
It helps stabilize the ribosome when it hits really tricky sequences like a string of prolines. And both spermidine levels and this hypusination drop with age. But if you restore them, you could extend lifespan.
And the loss of that modification, losing the locksmith, has a massive effect on the cell's ability to clean house.
A huge effect on autophagy, yeah.
Yeah.
The key proteins for autophagy, like the master regulator TFE, they have those tricky proline-rich motifs that absolutely require a hypusinated EIF5A to be made efficiently. So if EI5A isn't modified, TFE isn't made properly, autophagy fails, and all that cellular junk just builds up. The classic sign of aging.
And there's another player, the helicase DDX6, which is like a nutrient-sensing toggle switch for autophagy.
Exactly. DDX6 is so dynamic. When nutrients are everywhere, DDX6 actively degrades the transcripts for autophagy genes. But the second the cell senses starvation, DDX6 flips its function and enhances the translation of those same genes, quickly turning the self-cleaning system on.
Okay, so let's wrap this all together. Let's compare the acute stress responses we've been talking about to aging itself, which is really just chronic stress. What's the fundamental difference in how translation is managed?
The core difference, I think, is competence. An acute stress response is robust and it's specific. Stalling only happens where it's needed for a specific purpose, like repair.
Right.
Aging is chronic stress plus an insufficient response. Global translation does slow down in both cases, but the stalling you see in aging is generalized. It's random. It happens at sequences that are just chemically difficult to synthesize, like strings of lysine or proline.
Which suggests the machinery itself is just wearing out. It's not regulating anymore.
Exactly. It shifts from being this concerted adaptive slowdown to just being a symptom of system failure.
Yeah.
It's a detrimental consequence. The cell loses that ability to maintain the necessary balance.
It's just incredible. The last decade has unlocked this world of regulation, showing us how the cell uses everything from these core pathways to unique RNA flags and these specialized locksmiths to fine-tune protein synthesis under pressure.
But we have to remember, our understanding is still limited. I mean, most of these studies are snapshots. They're acute single stress events in a dish. Aging is hyperchronic. It's integrated across a whole organism.
Right.
The future is going to require these really high-resolution tools, like cryo-electron tomography, to actually measure translation happening inside intact tissues, to see how different cells fail in different ways as we get older.
So we've seen that slowing down translation can extend your lifespan, but if it slows down too much, it becomes a symptom of disease. Which brings us to our final question for you. What specific mechanism, if you could tune it perfectly, would let us keep that adaptive slow of youth without ever succumbing to the detrimental stall of old age? Something to chew on until our next deep dive.